Hey everyone, I wanted to show you this. I found this on the Alabama State Bar.org website. So it says, DNA analysis has long been labeled the gold standard of forensic testing by the court. While I acknowledge the scientific process of DNA testing as the pinnacle of scientific rigor when employed correctly by qualified scientists, certain aspects of the process can be shown to fall short of the objectivity necessary to be considered a scientific standard. To be considered a scientific standard, a method or technique must survive scrutiny and peer reviews. DNA analysis fits into the section of a crime laboratory called forensic biology. This unit is typically separated into two parts, serology and DNA analysis. Serology is the study of bodily fluids, and most forensic labs focus on identifying blood, semen, and saliva through screening, color change, and immunological tests. Body fluids are known to contain high quantities of DNA, making the downstream steps of DNA analysis much easier. Once a bodily fluid has been identified, the item is subjected to DNA analysis, which is composed of 5 steps: extraction, quantification, amplification, separation, and interpretation. The first 4 parts of this process are where the science happens. Scientists use chemicals and instruments to produce DNA profiles from the evidentiary samples and potentially known samples. These techniques are used throughout the scientific community in research and diagnostic labs. So the final step, interpretation, is unique to the field of forensics and where the process loses the gold standard label. Developing a mixture profile or a DNA profile with more than one individual is extremely common for most evidence submitted to forensic labs. Mixtures are the crux of the sexual assault matters, so much so that an extraction technique was developed to attempt to separate the male and the female portion of a sample, sometimes referred to as a sperm fraction, and the non-sperm fraction. Due to the increased sensitivity of these chemicals and instruments and increased requests to sample for trace DNA, note here, this is what completely drew me to this article. Not touch DNA, a topic for a whole nother conversation. I think that's really interesting that this author would put that in there, because why? Why, unless it's a problem? This author wants to be specific when they are trying to convey the message that they have about the DNA. So apparently it's touch DNA is not appropriate for this article. And I'd like to see that because I think we need more of that, that the touch DNA, not only should the public understand that that's not appropriate, it's not scientific, that also will help juries because, you know, I'm sure at some point we'll all be on a jury, maybe not, I don't know. But yeah, so it's good to know, especially because of these wrongful convictions and especially this Koberger case, this touch DNA, and that's exactly what prosecutors want. They want to implant into potential jurors' minds that the defendant touched a key piece of evidence. And how successful is that when you use the term touch DNA? And we still don't know if Brian was ever in the house, if he touched anything. We don't know that because the very nature of trace DNA is you never know when, and you never know how. I mean, sometimes you can know when and sometimes you can know how, but I think those are the exceptions to the rule. Most of the time, there is really no explanation. And the wrongfully convicted, there are lots of statistics on that. A lot of it has to do with faulty forensic evidence. And I just feel like if we're smarter about this trace DNA as a whole, as a public, then we can really see those wrongfully convicted statistics change. Yeah. So, note, not touch DNA, a topic for a whole nother conversation. Labs see mixtures of 2 to 5 individuals in a routine case work. Trace DNA refers to the amount of DNA present and does not imply an action that causes DNA to be present on that item of evidence. Wow. Let me repeat that again. So, trace DNA refers to the amount of DNA present and does not imply an action that causes DNA to be present on the item of evidence. Well, if that's the very definition of trace DNA, MPD, what are you doing? Jeez. I just don't see enough evidence. And then the evidence in the Koberger case that has been presented, there's a lot of questions about it. And it's also been stated in court on the record that the documentation isn't there for Brett Payne to make the claims that he's making about the cell phone and the video of the car. There's not one video. Although he did say everything that they've had was turned over to the Moscow Police Department or the prosecutor's office, and or both. So if they did collect the Monday's Machine or the Red Star coffee, did he say that was turned over but nobody can find it? Well, no. Anne did say that Monday's Machine was turned over and nobody can find it. So they have it. So you're missing key documentation that is required to make the allegations that you're making against Koberger. And you're also missing a video that you say you have. So, I mean, that's not even all of it. That's just the beginning of it. And then you have this definition of trace DNA. Trace DNA refers to the amount of DNA present and does not imply an action that causes DNA to be present on that item of evidence. Interesting. And, there is paperwork out there that says trace DNA should not be the only evidence you have against somebody. I think that's actually the DOJ guidelines, but we can read that later. Anyway, so for scientists, sensitivity is a double-edged sword. On one side, we have more data, which means we can potentially use this data to identify more perpetrators or provide more information to investigate the court. More data also equates to better statistical calculations that provide more weight to our conclusions. Yes, like I was saying yesterday, when you speak about matching DNA, you're speaking through statistics. So there's always a number attached to the percentage you match the source DNA. This data helps analysts assist in revealing more of the truth using science. On the other side, it leads to many questions about how well we can confidently and accurately identify an individual. Mixture interpretation, especially of 4 or more individuals, has always been a subjective aspect of the forensic biology process and fails to survive adequate scrutiny by peers. As chemistries and instrumentation sensitivities continue to become more sensitive, the subjectivity of interpreting the results increases. The subjectivity is well documented. One such study was completed by the National Institute of Standards and Technology, NIST. Oh, that EWG working group came from that same group. Okay. Which used a report on findings in 2018. In the study, they compared two interlaboratory studies named MIX-05 and MIX-13. These studies reflect the years in which they were performed, 2005 and 2013, respectively, and compared the protocols and procedures of labs across the United States on how they interpreted mixture profiles. The labs were given multiple DNA mixture profiles of 2, 3, or more persons, meaning it could be 3 or 4 or 5 or more, and known standards to compare with these profiles. Then each lab provided conclusions based on their standards and operating procedures. This was great. I briefly read through this earlier. This is like everything right here. Okay, so if you're still determining whether subjectivity is a part of DNA analysis, here's your answer. For a single lab included in this study, when the responses were reviewed for one scenario in MIX-13, 50% effectively said, I don't know. Inconclusive, 30%. When responses were reviewed for one scenario in MIX-13, 50% said, I don't know. Inconclusive, 30% said, he's not here. Excluded. And 20% said, he's not only in the mixture, but I can exclude greater than 99.9% of the population. Match. This implies that presenting favorable or unfavorable DNA results to a person of interest may depend on which in the lab processes the evidence. Should this be considered a gold standard? This study expresses that this study does not provide a full view of the day-to-day activities within a lab, and the results may not have undergone normal coursework requirements, such as undergoing a technical review or multiple analysts. How can we move on solving this issue? If there is no way to establish a set of protocol for each and every forensic lab using different chemistries and instrumentation, the best answer we have right now is probabilistic software systems. We are encouraged by the development of probabilistic software systems. PG software uses biological modeling, statistical theory, computer algorithms, and probability distributions to infer genotypes, DNA profiles, and likelihood ratios. The software uses the files generated by the separation step of DNA analysis, or all the DNA that is used to produce a picture of a DNA profile called the electrophenogram, and deconvolutes the profile. The deconvolution, or process of determining the possible contributors in the profile, occurs using a statistical method called Markov chain Monte Carlo, MCMC, that relies on random sampling to find the genotypes that best explain the composition of the mixture DNA profile. This software removes some of the subjectivity of interpretation by employing validated biological modeling that can include specific data from the lab. However, this software processes a sample twice. It will not provide the exact same answer, but will be clustered around the best fit answer. Currently, two well-known companies have produced software that forensic labs are taking advantage of in casework. They both work the same mathematical principles described above and can be deconvolute mixtures of more than 5 individuals. Both have been presented at trials in multiple states in the U.S. and accepted in admissibility hearings. PG software allows analysts to harness all the data produced in the scientific process and apply well-studied and mathematical sound calculations to provide a more objective, unbiased, and accurate view of mixture profiles. Although this software is a move in the right direction, it is a tool that must be used aptly and after proper training and validation have been completed. As an attorney, why should you care about this study, PG, and the subjectivity in forensic biology? It comes down to two essential things: voir dire and cross-examination. The findings of this study specifically highlight the need for improved training and striving for consistency. When preparing for trial, you must obtain as much information about the analyst as possible, including their training record, proficiency tests, and personnel files, along with the entire case file. If they have issues with their casework, it will be present in the documents. If their training record states nothing about mixture interpretation in the past 2-3 years, you need to press them on what they are doing to prevent this subjectivity. Ask them if they have participated in any training in PG and if the lab is moving forward to validate and bring on this software for casework. Don't be afraid to get them to dig deep into their conclusions during cross-examination. Simply read the report to the jury. Simply reading the report to the jury provides just the tip of the iceberg of the data they collected. Remember, that report is the result of 5 steps. All forensic scientists should be able to answer questions about their entire process. Furthermore, they should be up to the task of relaying this information in a manner that can be understood by you, the court, and the jury. Even if the lab does not employ PG, they should have a basic knowledge of what this software is capable of and what information it could provide. That is their duty when they climb in the stand and are deemed expert witnesses. As the attorney, you must ensure they are held to this standard and reveal this subjectivity so that a jury is fully aware of the standards in forensic biology. Although subjectivity will never be fully removed from the forensics field, all criminal law system participants must strive to ensure we decrease it as much as humanly possible. Isn't that interesting? So this article also references that you need to obtain their training record, or as much as the information as possible, their personnel files. And that's exactly what Anne was requesting. And I remember hearing people out there and be like, oh, that's just above and beyond. Like, she can't ask for that. Well, I don't know about that. I feel like attorneys do ask for that. And apparently it is needed to help the jury with their decisions. And so when you keep certain information away from the jury can't do its job. The jury has a job to do. And it is the responsibility of, the attorneys to make sure that they are defending their client and they're getting all the information they need so the jury can hear as much as possible. And it just seems that the state of Moscow is trying to prevent information, which is very strange. And then, there's also people out there who are for the state, and to me, it sounds like they agree that the jury doesn't deserve the information. Which that's really weird. Anyway, so, and does it say who this was written by? Samantha C. Spencer, and it was written February 8th, 2024. From the Alabama Lawyer, Cross-Examination of the Forensic Gold Standard for DNA Testing. Awesome. Great article, Samantha. Wow, so it was written by an attorney, maybe. She really knows. I mean, this is really well written.